Hello, I'm Mark Carr. I'm a marine ecologist in the Department of Ecology and Evolutionary Biology here at the University of California, Santa Cruz. I'd like to share with you some of our research that's revealing how populations of rockfishes along the coast of Central California are actually connected to one another through the movement of their young. Unlike most land animals, whose young remain with the parents that produce them, the vast majority of species that live in the ocean produce young in the form of larvae, little transparent individuals that are adapted to be carried away by ocean currents to replenish populations somewhere else. For example, the larvae born from female kelp rockfish that live in kelp forests along the Central Coast are carried away by ocean currents. And if they survive, and if those currents deliver them back to shore, they'll settle to replenish populations in kelp forests somewhere else along the coast. Likewise, future generations of that population of adults is determined by the delivery of young that are produced in another kelp forest elsewhere along the coast. This is a crucial process for marine species because it determines how populations grow in size, and it also influences how populations contribute to the replenishment of one another. It also has very important conservation implications because the young that are produced in a marine protected area can actually replenish populations that are fished somewhere else along the coast, and it also determines the growth of the populations within those protected areas. Until now, it's been very difficult to determine where young travel along the coast to replenish other populations, and how the ocean currents actually determine where they travel to. To answer this, we've brought together a team of ecologists like myself, geneticists like Dr. Carlos Garza at the National Marine Fisheries Service, and oceanographers like Dr. Chris Edwards in the Department of Ocean Sciences at UC Santa Cruz.
My name is John Carlos Garza, and I'm a research geneticist at the Southwest Fisheries Science Center in Santa Cruz, California. We're studying the movement of larvae of kelp and black and yellow rockfishes, which are two species that live in the kelp forests of Central California and are important in recreational and commercial fisheries. Our study is focused on populations in Carmel Bay and around the peninsula of Monterey. We have developed and are applying a novel genetic technique that allows us to compare the genes of adult and young rockfish as they're settling into their new kelp forest homes. We can get a complete genetic profile from these fish from a small piece of tissue taken from their fins without harming them.
My name is Diana Betcher. I'm a PhD student at UC Santa Cruz, and I work on larval dispersal of rockfishes with Carlos Garza. What we do is we extract the DNA from the fin clip and put it into solution. So then you have this clear solution with the DNA. What we're doing, as Carlos mentioned, is selecting these genes. And in order to look at those genes in the parents and the offspring, we have to amplify that gene in all of our rockfish samples.
This genetic technique allows us to compare the genetic profiles of all adult and juvenile rockfish that we sample and determine who is related to whom. If we find a genetic match between adult and juvenile rockfish, then we know how far those young fish have moved during their lifetime. That's because we know where they settled, where we sampled them, and we know where they were born, where we sampled their parents, because fish of these two species move very little after they settle into their kelp forest homes. To sample the young fish, we deploy collectors that mimic kelp forest habitat and that the young fish settle into. We check these collectors each week to obtain samples from the young fish that have settled at that site in the previous week. Employing this technique, we've now found parent-offspring and sibling matches that indicate that rockfish populations simultaneously replenish themselves and contribute to other populations along the coast. This means that populations in marine protected areas contribute both to maintaining themselves and also to maintaining populations outside of these protected areas that may be subject to fishing. This is a win-win for conservation and fisheries, and something we're very excited to discover.
Hi, I'm Chris Edwards, an oceanographer in the Ocean Sciences Department at UC Santa Cruz. By knowing where larvae were born and where they settled, we use computer models of ocean currents to understand how they were transported from their birthplace to their destination.
I'm Yana Lowe. I'm a graduate student here at UCSC. I am studying the ocean currents and how they transport larval rockfish. This is a view of the computer model we built of the ocean currents off the US West Coast. We are located here in Santa Cruz, and this study takes place in southern Monterey Bay and Carmel Bay. This simulation shows the sea surface temperature during the spring and summer months. These blue regions show cold water near shore. Later in the summer, the water warms, and we see that here in the map with all these red colors.
By knowing how long the fish larvae drifted with ocean currents and when they were delivered to their settlement collectors, we can examine the ocean conditions that may explain why they were transported from their release to their settlement, as well as the conditions that they may have experienced along the way.
In this simulation, imaginary fish larvae are represented as black dots that are released in Carmel Bay. These larvae are moved by ocean currents and go in a variety of directions due to changing winds and nearshore currents.
The genetic comparisons show that some young born in Carmel Bay return and settle in Carmel Bay, while others are carried north to Monterey Bay. Likewise, some young born in southern Monterey Bay remain in southern Monterey Bay, while others are transported south to Carmel Bay. The ocean is very dynamic, with quick changes in temperature and in current direction. Our models suggest that these rapid changes help explain how larval rockfishes ended up where they were collected. The more we learn about how and where fish larvae are transported along the coast, the better we'll understand how populations can help replenish one another along the coast, and how populations in marine protected areas can help replenish fish populations, as well as those populations in other protected areas.
We appreciate this opportunity to share our research with you, how it's advancing our understanding of marine ecosystems along the coast, and also informing conservation efforts, like the design of networks of protected areas. Thanks for watching.